Okay, we're going to return to Objective 4. Remember that hormones are just chemical messengers that the endocrine glands secrete, and they travel in the bloodstream, and so the amount in the blood varies minute to minute, and they regulate the functions of other cells. Now there's two main types of hormones. This is Objective 6, classified by their chemical structure. There's the amino acid hormones and the steroid hormones. The amino acid hormones, obviously, are made of amino acids. If it's one amino acid, it's called an amine hormone. If it's multiple amino acids, it's called a peptide hormone. And if it's a complete protein, it's called a protein hormone. These are usually hydrophilic, so they're going to bind to receptors in the plasma membrane. Now the steroid hormones come from cholesterol, and these are hydrophobic hormones, so they're going to bind to receptors that are in the cytosol or the nucleus. Now, Objective 5 is where we are. We're going to look at how hormones are transported through the blood. They can be unbound and travel freely, or they can be bound and attached to plasma protein carriers. If they're free hormones, they're small, they're amino acid based, they're hydrophilic, and so they can travel easily through the water-based plasma in the blood. However, they don't last very long. There's a few of the hydrophilic hormones, like growth hormone, that are bound to proteins. Now the bound hormones will form these complexes by binding with proteins in the plasma. These are usually hydrophobic, so they don't do well with the water in the plasma. So binding with the proteins helps them with that, so that they can be transported safely in that watery plasma. This provides us with a reservoir of hormones that are released when needed, and it also helps to extend how long the hormone lasts. It extends their lifespan, which we'll talk about later. Now moving back to Objective 4 again, hormones only affect specific cells that we call target cells, and those target cells have to have a protein receptor for the hormone to bind to. And they fit together like pieces of the puzzle. If the receptor is shaped a certain way, the hormone has to fit in there, in that receptor. So it's very specific. It won't just accept any hormone. And the receptors are either embedded in the plasma membrane, or they're in the cytosol or the nucleus. Now moving to Objective 5. Where the receptor is found depends on the chemical structure of the protein. Is it hydrophilic or is it hydrophobic? If it's hydrophilic, it has trouble getting through that phospholipid bilayer. And so it's going to bind to receptors on the target cell's plasma membrane. If it's hydrophobic, it's got no problem going through that phospholipid bilayer. So it's going to bind with receptors in the cytosol or the nucleus. Cells can control how they interact with hormones by changing the number of receptors that are displayed for the hormone to bind to. Upregulation occurs when they produce and display more receptors. And so that increases that cell sensitivity to the hormone. It raises the hormone level in the blood. Downregulation occurs when the target cells decrease the number of receptors for the hormone to interact with. There's nothing for them to hook up with, so they can't cause an effect in that cell. And this can happen if you have exposure to a hormone at a high level for a long period of time. Now the hydrophilic hormones, how they work is they are considered the first messenger. And we've already said they bind to the receptor in the plasma membrane. That receptor activates a peripheral protein on the inside of that membrane, right where it joins, right where it is with the cytosol. G protein would be an example of this. That G protein activates an enzyme, and that enzyme catalyzes the making of the second messenger, and that will cause changes to happen in that cell. Adenylate cyclase is an example of an enzyme that the G protein activates to form the second messenger. And the second messenger it forms is cyclate adenosine monophosphate. And that will cause changes in the activation of protein kinases. And that makes it so that you can transfer a phosphate group from one ATP to another molecule. Now the hydrophobic hormones, that hormone has no trouble crossing the plasma membrane. So it's going to interact with receptors in the cytosol or the nucleus. And it's going to form a complex with its receptor. And that complex will change the rate of protein synthesis.